A very good afternoon to Dr. Yu. I'm SURITHA, and today my groupmates and I will be presenting about Ascaris lumbricoides. To start off, Ascaris lumbricoides is known to be the largest intestinal roundworm found in the world, and it affects people of all ages, starting from children to adults. The main characteristics differentiating male and female is through the size. Females are known to be a lot bigger. The pathogenesis of this infection starts off with ingestion of the eggs. So children of age 3 to 8 years old are more prone to this infection because when they play in the playground with toys and when they place their hands into their mouth, the ingestion of the eggs begins, and this is where infection can start. Another way is through consumption of contaminated food or water. So the picture on the left here is the life cycle of Ascaris lumbricoides. So it starts off with ingestion. After ingesting the eggs will move down into your small intestine, and where it will also be migrated into the bloodstream as well as the lungs. So in the lungs, it will be coughed out and swallowed back down into the small intestine, and this is where it can grow. When it grows and it becomes a larva, it feeds onto the nutrients of the host. So then, some of the larvae will be excreted through feces and others remains in the body, and the whole cycle repeats again. After the stool samples are being collected, specimen containers are labeled with the patient's name as well as identification, card number and other clinical information, and then they are sent to the laboratories for inspection. After the lab technician has received the stool sample, it first undergoes macroscopic examination, which is visually looking at the stool sample for any blood, mucus or other abnormalities. It then undergoes concentration technique like sedimentation or flotation, where the eggs of the stools are being viewed under the microscope. And lastly, if eggs are detected, they are being confirmed and reported to patient, and further analysis will be done. I will now pass the time to my friend Loges to talk about the epidemiology of Ascaris lumbricoides.
Hello and a very good day. I am LOGESWARY NADARAJAN, and today I am going to talk about epidemiology of Ascaris lumbricoides. My first point is geographical distribution. Ascaris lumbricoides is found predominantly in tropical and subtropical regions, including parts of Africa, Asia and Latin America. Within these regions, endemic areas often coincide with poverty-stricken communities where infrastructure for sanitation and healthcare is lacking. Next, prevalence. Ascaris lumbricoides is particularly common in areas with poor sanitation and hygiene practice. Regions with inadequate sewage system, limited access to clean water, and overcrowded living conditions are more likely to have higher rates of infections. Apart from that, risk factors. Beyond socioeconomic factors, certain behaviors and environmental conditions increase the risk of Ascaris infection. Lack of access to adequate sanitation facilities such as toilets or latrines increases the likelihood of fecal contamination of the environment. And also, overcrowded living conditions common in urban slums and impoverished rural areas facilitate the spread of infections. Based on the figure, in America, the prevalence of Ascaris lumbricoides infection varies widely depending on the region and socioeconomic factors and access to sanitation. In some endemic areas of Central and South America, prevalence rates of Ascaris infection can range from 20% to over 70% in certain population, particularly in rural and low-income communities. For instance, a study conducted in Mexico reported an overall prevalence of Ascaris infection of around 7.5% among school-aged children. Not only that, most affected age group is children, particularly those living in impoverished condition with a limited access to clean water and sanitation facilities are often the most affected by the Ascaris infection. This is primarily due to their behavior such as playing in the contaminated soil, poor hygiene practice and developing immune system that is more susceptible to parasitic infections. So factors that contributing to infection is poor sanitation, polluted soil and low socioeconomic level are the main causes of Ascaris infection.
So some of the symptoms of Ascariasis that you can be found in the large intestine and small intestines are intestinal obstructions in the liver, gallbladder, pancreas, cholecystitis, appendicitis, acute pancreatitis, and if Ascaris actually moves to the lungs, it can cause asthma, and when you cough, you'll be able to see some of the Ascaris inside the oscitum. And also, these symptoms can be detected by the advanced diagnostic methods such as MRI, CT scans, X-rays, and radiographs. The pros of these are they are able to detect the abnormalities, but they can't actually detect what kind of specific helminths, specific nematodes, cestodes, because the symptoms are shared among many other parasites. That's the cons. And then we also have the conventional diagnostic methods, which are much more specific, such as the fecal examination and also the complete blood count. Complete blood count consists of eosinophilia, because eosinophils are produced by the body to counter the parasites, and in the fecal diagnostic test you'll be able to see many parasites inside the stool. But the cons of this is that it takes a longer time and it's harder to see. If you don't have a keen eye, it's hard to see. The pros are that you can specifically know what kind of helminth it is and give the correct medication to treat it. Thank you.
Let's see the conclusion. Choosing between conventional and model methods for controlling Ascaris lumbricoides depends on various factors such as effectiveness, accessibility, and sustainability. Conventional methods such as health education, improved sanitation, and deworming programs have been foundational in combating Ascaris infections for decades. In conclusion, while both conventional and modern methods have their merits, and pragmatic approach involves leveraging the strengths of each within the context of local epidemiology and available resources.